The broadcast is now starting. All attendees are in listen-only mode.
Hello, everyone, and thank you for joining us today. You've logged on to the Nelson Labs webinar, Packaging Validation 101, Part 1. You can now register for Packaging Validation Part 2, Process Control, which will be on June 3rd. You can find that link on the Nelson Labs website. It will also be emailed out to you after this event. Now, let's get into a few housekeeping items. If you missed one of our webinars or would simply like to refer to one, you can always find them on the Nelson Labs website on the On-Demand Webinars page listed under Education. You can receive notifications for upcoming live and on-demand webinars by liking Nelson Labs on Facebook or following on Twitter or LinkedIn. Nelson Labs is a leading global provider of laboratory testing and expert advisory services for medtech and pharmaceutical companies. The company performs over 800 rigorous microbiological and analytical laboratory tests across the medical device, pharmaceutical, and protective barrier industries. The experts at Nelson know every test matters and requires solutions to complex problems to improve patient outcomes and minimize client risk. So let's get started. Today we're joined by Jennifer Gigi, an expert technical consultant from Nelson Labs. Jennifer has over 25 years of medical device laboratory experience. She has worked in the microbiology, bioburden, IDs, and packaging sections at Nelson Labs. Jennifer has experience with various microbial tests, including microbial limits, plate counts, biological indicator population verification, organism IDs, and bioburden tests. Jennifer has spent over the last 15 years working in the packaging group and as one of the original packaging group members at Nelson Labs. Jennifer was heavily involved in validating all the packaging test methods and equipment and writing test procedures for the packaging tests performed there. Jen is a participating member of the ASTM F02 committee for packaging and is registered as a specialist microbiologist with ASM. Now beginning today's presentation, I'm going to turn the time over to Jen.
Thanks, Mike. Well, we've got a lot to cover today, so let's get going. All right, in this webinar today, let's see, here we go. Here's our agenda. We're going to start with an introduction and overview of ISO 11607, which is the primary packaging validation standard. And next we'll cover sample size considerations and go through some basics on how you can calculate a sample size for your validation. Then we'll discuss the validation protocol and what it should include. And then we will be talking about developing the test plan and the protocol, including selecting appropriate tests to demonstrate the strength, integrity, and microbial barrier properties of the packaging, and selecting time points and aging conditions. Then we'll explore some family grouping and worst case considerations that can be helpful in keeping validation costs lower when you have multiple products that are similar. And finally, we'll conclude with some helpful tips and best practices for creating the validation samples and sending them to the lab for testing. ISO 11607 was first published as a standard in 2006. Before this, there wasn't really a documented process for running a package validation. As such, companies each had to invent their own wheel, so to speak, and come up with their own validation plan. Some of these validations were very robust and others not so much. Since its publication, the world has very specific guidance on what a packaging validation needs to include and how to do it, though the standard's very open and there's a lot of options available for validations tests. ISO 11607 is divided into two parts. Part 1 is how to make and validate sterile barrier packaging, and Part 2 is how to perform process validation. Today we're focused on Part 1, but we'll be covering process validation in a separate webinar in the near future. There's also guidance in the document on how to interpret and use ISO 11607, and this is ISO 16775. I would recommend purchasing both for your company library. They're essential guidance documents to have if you're making sterile medical devices. Perhaps the most common question that we get around packaging is, how many samples do I need to test? Now, that's a very loaded question because sample size depends on a lot of different things. Try to answer this question. Let's start with the guidance we have from ISO 11607. Both 11607 and 16775 are using the same phrase in regards to sample size, which is statistically valid rationale. We also know that a risk-based approach is important because many of the factors that play into sample size are risk-related. Let's look at those factors next. Higher sample size provides more statistical confidence, but the more you test, the more it costs. Balancing this cost versus risk is the tricky part. Now, a good statistician will tell you that the relationship between sample size and risk is complicated, and it's not linear, meaning that I may get the same statistical confidence with a range of sample sizes. So the goal should be to aim for the lowest sample size that gives you the best statistical confidence. In addition to cost, you should look at the process variability. A tight process would allow for a lower sample size. A more variable process would require a higher sample size. The process controls are also important. If you have midstream controls and checks, you may be able to justify a lower sample size of the final product. The confidence level plays into risk. If the risk is low, then you can justify a lower confidence interval, which in turn gives you a lower sample size. Are you sampling using an even distribution within the group, or are all the samples coming from relatively the same location? A more even distribution will be more likely to catch random defects. How often are you sampling? If the frequency is high, you may be able to justify a lower sample size because many small data sets might paint a better picture than a single larger data set. Sometimes you're limited on the number of available samples. Maybe your lot size is small, or the samples are part of a small group that has been real-time aged. The test you choose also makes a difference. Data, which includes a measurement, can be statistically evaluated to see how much variability there is in the set. This goes back to variability within the population. And you may be able to decrease your sample size and/or testing frequency if you have historical data showing that the process has been under control for a long time. The method sensitivity and the expected % defect in the lot are also considerations. If you're expecting a high defect rate, then a smaller sample size would detect it. But if the defect rate is low, then you would need more samples to have a chance that one in your set is defective. If the expected defect is easily detectable, then there's a higher probability of finding it. If you're using a sensitive method that has a high probability for detection, well, maybe you can use a smaller sample size. Hopefully, you're getting the picture that it isn't as simple as picking a number off a chart. There are a lot of variables that should be considered and play into sample size decisions, and it really depends on why you're sampling and what you're testing for. And my final thoughts are that the actual number is really not as important as the why behind it. And you can't say that any single number is the gold standard. This slide has the most common approaches that we see for determining sample size for packaging validation. Certainly, there's no one size fits all, and there are multiple ways to do this. Just a word of caution. If I gave you a bullet point list on how to bake a cake, grab the ingredients in the recipe, mix them together, bake them at the time and temperature in the recipe, then would every cake be identical? Well, probably not. So while this is the basics, just remember that there are a lot of moving parts behind the scenes, and the most important component is the why behind your selections. The first option we look at is the risk priority number. In this approach, you develop a risk scale, which most companies already have. Then you look at what a packaging failure looks like in relation to that risk scale by answering this question. If I have a packaging breach, what does that mean in terms of risk? Once you have a good idea of the risk inputs, then you can calculate the risk priority number, or RPN. Remember to justify why that RPN is appropriate for your situation. And then from an RPN table, and you can find a bunch of them out on the internet, you can correlate the RPN to a corresponding risk level, maybe high, medium, or low. And that will usually give you a matching confidence and reliability interval. The last step is to convert the confidence and reliability interval to a sample size. And we do that by using maybe a non-parametric binomial reliability demonstration test table. Again, you can find a number of those out on the internet, and some have allowable failures and some don't. And so you want to select an appropriate table based on your situation. The next method on our list is the acceptable quality level, or AQL. Now this is based on ISO 2859-1, and you start by determining your lot size. Generally, as the lot size increases, your sample size will increase. Then you need to define what the nonconformity is. Well, what is that defect that you're looking to find, and how will you find it? This plays into method sensitivity, nonconformity detectability, and the expected defect rate. Then you decide on your AQL, which is the amount of risk you're willing to take. The AQL represents the allowable defect rate before a lot is rejected. Can you allow 1 in 100, or is it 1 in a million? Each has a corresponding AQL number. If the risk of that nonconformance is low, meaning if you do have a nonconformance, it isn't the end of the world, then you can have a higher AQL, like a defect rate of 1 in 100. If the failure would pose a high risk to patient safety, then you're going to need to get that AQL as low as possible, maybe 1 in a million. Now you pick the sampling plan table from the list that's most appropriate for you. Then you pick an appropriate inspection level based on the amount of risk you can tolerate. Both these selections should be justified and explained. And then you use the lot size and inspection level to get a sample code letter and match that to the sample plan table. And voila, you take your AQL and you come up with a sample size. The third option would be to hire a statistician who can go through all your individual risks and calculate a custom sample size using statistical formulas. Now, this is probably only something I would do if you already have a statistician on staff, or if you're looking for a really specialized sampling plan for QC testing. For package validation testing, the other options are generally sufficient. Nelson Advisory Services, which is the Nelson Labs consulting group, can write these packaging sample size justifications for you, and we do write a lot of them. So if you're interested, just let us know. I want to touch briefly on attribute and variable data, because this relates directly to packaging validations. Attribute or qualitative data is pass or fail. And so you don't get a numerical value that you can analyze statistically. Tests for package integrity, such as bubble emission or dye migration, are pass-fail tests. Variable or quantitative data is a measured result, and you can run statistics and see how uniform your sample set is. The package strength tests, like peel strength and burst strength, generate quantitative data. Generally, attribute data should have a larger sample size because of the nature of the data, and we usually recommend addressing these two types of data separately in the sampling plan, though that's an individual decision. It all starts with a plan, or at least it should. If you don't have a plan in place, then how will you know that what you're doing and if you're successful at doing it? The validation protocol is the written plan that tells the story of your validation. You remember the lessons from elementary school about good storytelling? You start by asking the 5 W questions of what, where, why, when, and how, and then you write it down. For a packaging validation, you need to say what you're doing, why you're doing it, and the why is usually the most important part. How you will do it, when you will do what, and where you expect the outcome to lead you. In this case, you expect to meet all your acceptance criteria and successfully demonstrate that your sterile barrier system is effective until the point of use. If you just toss a bunch of data into your submission file, the FDA or other notified body is not going to accept it. Not that they don't understand what the data is or what it means. No, they're very familiar with what a packaging validation should include. The point is, they want to know that you know what it all means, which is why the protocol that tells the story and sets up the rules for acceptance is so important. It shows that you understand, that you have a plan, and you aren't just winging it and hoping for the best. Then you need to follow your plan and explain the outcome. These are some key elements that should be in your protocol that answer those 5 W questions. Describe the device in the packaging system. What is your intended sterile barrier? How is the packaging system set up to protect the package? It's important to describe in detail so that the original sample is well documented for later when changes are made, or so the FDA can verify that you actually validated what you said you did. Lay out your test plan, including what tests you're going to perform and why those tests are appropriate, when you're going to do the tests, and those are the time points for the study, and how many samples you'll run for each test. How you'll age the samples, both real-time and accelerated if you're doing accelerated, and what your acceptance criteria is and why that criteria is important. The test methods usually don't define what result value is acceptable, so each individual needs to decide what a good minimum value is for their specific product. A good example is the strength acceptance criteria. The minimum seal strength value is what's needed to keep the package closed, and that can vary depending on how heavy your product is or how much stress is being put on the seal, which can be related to product weight and our pressure vacuum changes during sterilization and air transport. Another quick plug for Nelson Advisory Services. We offer a complete validation protocol for purchase that's been customized for your product and meets the requirements in ISO 11607. So contact us for that if you're interested. Per ISO 11607, the sterile barrier system needs to be evaluated through all intended processes. This includes sterilization, handling, distribution, and storage. The device should be in its final packaging configuration for testing, and it should be created under worst-case conditions. This means that a worst-case sterilization is preferred, since sterilization can affect packaging. And if possible, it's best to seal the packages at the worst-case sealing or low settings versus the nominal. When a worst-case package passes the validation, you have an extra measure of confidence that anything atypical that happens during routine production will be covered within the scope of what you've validated. There is no single packaging test that can definitively say that a package is good. So we look at 3 different aspects that together give us a more accurate picture of the package performance. These are strength, integrity, and microbial barrier. My 3-legged stool pictured here would be wobbly if one of the legs was missing. Likewise, the packaging validation is not as solid without the multi-property evaluation. I also like to compare it to triangulating a position using GPS. One point gives you a wide radius or a general idea of your location. A second point narrows that location down, but a third point can pinpoint with far greater accuracy your exact location. ISO 11607 has a very long list of tests that can be used to evaluate many properties, and if none of those listed options appeal to you, then they allow you to create a custom method as long as it's validated. Nelson Labs offers the most common tests used in industry in each of these 3 categories. This table shows our offerings for strength, integrity, and microbial barrier tests. Each of these methods has pros and cons, and some are preferred over others in certain situations. For a packaging validation, you would select at least one test from each of the 3 columns. Here's the overview of a basic validation plan. You start with the final product in the final packaging and sterilize it using the validated sterilization cycle, preferably a 2x or double sterilization to get to that worst case condition. Then the boxes are run through a distribution simulation, and then the boxes are inspected for damage. If there's a distribution failure, let's say a box crushes in the compression test or splits open in the drop test, then you need to investigate, determine the severity and potential impact. If the severity is low, you may be able to proceed to the next step. But if the severity is unacceptable, then you need to redesign the protective packaging and start over. Step 2 is the baseline testing on the sterile barrier system for strength, integrity, and microbial barrier tests that you selected from the table. If you pass baseline by meeting all the acceptance criteria in the validation protocol, then you move to the next step. If you have a failure at baseline, you need to stop and investigate to determine the cause and severity of the failure. Then, based on what you find, you either need to do additional testing to confirm the original failure was an anomaly, or you go back to the beginning and redesign the packaging and try again. Most of the failures that will occur in the validation are going to be found at baseline. So this is a key gate to get through. If baseline testing is good, then the probability of failures after aging is low. If you have problems in baseline and you struggle with additional testing, you'll probably encounter similar problems after aging, so keep that in mind. As difficult as it may be to scrap the validation and start over, both in terms of time and cost, in the long run, it may be the best alternative. As bad as it is to start over at this point, it's far worse to do so after aging. And that brings me to another related thought. Though it's not specified or required, doing some preliminary testing of your packaging is a great idea to confirm your design is sound. A packaging validation usually costs maybe $20,000 to $50,000, depending on how complex you make it. And it usually takes 6 months to a year to get to the point where you can submit data to the notified body. Now, that's a pretty big investment, and it only makes sense to do some R&D work up front. This doesn't need to be anything fancy or even anything official. It could be as simple as tossing the box around the office, dropping it off your desk, or driving around with it in your trunk, or maybe sending it via courier to another location and back. Then open it up and look inside. Have the contents shifted? Do they look damaged or like new? And what does the box look like? You can get some good unofficial information very early on before committing to a formal validation study by doing something like this. And it really makes a lot of sense, especially if you're working with an unproven design. You can also send samples that you've beaten up and stressed out over to the lab for bubble testing to see if they'll pass an integrity test even when they look awful, as a preliminary check before starting the validation. Now, let's talk about aging. At this point, you've shown that your packaging, when shiny and new, works as expected, and now the final step is to prove that the packaging is still doing its job at the end of its expected life. Real-time aging is required. That means that the product is stored in the expected storage conditions, which is usually room temperature for most medical devices, but it may be refrigerated or frozen. And we need to do that for the expected labeled expiration date. If you want to claim a 5-year shelf life, it will take 5 years to age the product. And that's not really great because you want to be selling your product. So you can use accelerated aging at temperatures up to 55 C to speed up the process. It only takes 7.5 months to get a 5-year accelerated age product. You can submit to the notified body with just the accelerated data, but you're required per ISO 11607 to start the real time within 3 months of the start of the accelerated portion. And this is really just to ensure that the real time isn't forgotten. The aging time points are chosen by you and based on what you want your label claim to be. You must test baseline or time 0 and the final time point. But sometimes people include an interim time point as an insurance policy in case something unexpected happens at the final time point. Validation costs increase with longer final time points and with additional interim time points. After aging is completed, You repeat the same testing that was done on baseline for strength, integrity, and microbial barrier. And if you meet the acceptance criteria in your protocol, then you've proven that the sterile barrier system remains intact and effective through the expected expiration date. Here are some test method key points to remember. Any test method used in the validation needs to be validated. We prefer to use the methods on the ISO 11607 list because they're standard methods that have been subjected to some regulatory scrutiny. Reviewers and auditors are familiar with them as there's a history of use in the industry, and most were tested in multiple labs prior to being published to document the ruggedness and reproducibility of the method. Just because the method's from the standard, though, doesn't mean that it doesn't need a validation. Each lab performing the method needs to run a validation using their equipment, facilities, and personnel to demonstrate that they can perform the method internally at the same level as was established in the interlaboratory study done by the standards group. Some things that should be included in the method validations are ruggedness and reproducibility to show that the test can be performed by different people at different times, and you can still get the same result. For methods with a published sensitivity, you should include a demonstration that your lab can detect the defects in the published sensitivity range to the same level as published in the standard. And one last note, some test methods are really a process, like aging and distribution, that are really just preparing the samples for evaluation using a different test. As such, you wouldn't validate the method, just the equipment used to perform the process to show that the process is going to be the same each time. The first leg of the validation is the transportation distribution simulation. This is a process meant to simulate the expected hazards that a package will typically see during shipping. Most shipping vehicles are not climate controlled, and packages will experience changes in temperature and humidity depending on the season and geographical location. Boxes in large stacks will experience compression from the load on top of them, since the trucks are filled from top to bottom to maximize space. All boxes in the truck will experience vibration and shock forces as the truck travels down the road. While the intensity of real-world hazards will vary, the typical hazards and expected severity has been translated into a uniform, reproducible laboratory simulation, which, when passed, gives you a measure of confidence that your package should arrive to its destination and not look like my picture here. Yes, this box was an actual test sample that was received in this condition. Some of the boxes on the pallet looked like this one, but the majority were just fine. Since distribution simulation is for typical situations, you could still occasionally get an adverse event if there was an extreme hazard that occurred, which seems to be the case for this poor box. Distribution needs to be the first step in the validation because this is where all the damage usually occurs. And as part of the simulation, each hazard is run and the shipper box is inspected after each one. But it's really the damage that's happening on the inside to the sterile barrier system that really matters to us. And that's what we're going to check for in the next validation step. Let's look at the typical distribution hazards. First, we start with climatic stressing. This is where we simulate those temperature and humidity extremes. We usually cycle through an extremely cold, a hot and dry, and a warm and wet simulation. We simulate shock by dropping the box on edges, faces, and corners onto a hard, flat surface. We simulate compression by squeezing the box between two plates to a specified load. We simulate vibration in long-haul trucks and airplanes using a random vibration frequency table, and we simulate intercity trucks using a shaker table. We test impact strength using a rounded probe, and altitude is simulated using a vacuum chamber. Those are the hazards. Now let's watch a quick video of a distribution simulation that's been abbreviated so we can see how each of these tests work in action. So here's the compression test, and we're going to do the drop test. That's our shaker table. That's the vibration table. Then we're going to drop them one more time at the end. And we may do a concentrated impact test or an altitude test, depending on how the sample is constructed. After distribution, we need to test the sterile barrier system for those 3 attributes. Let's start with the strength testing. The strength component ensures that the package is sufficiently sealed to remain sealed and thus sterile until intentionally opened. The 2 most popular options are seal, peel, and burst. Both these tests generate variable data or a numerical value, and for both, you need a minimum value as the acceptance criteria. The strength minimum should be a value that's within the expected sealing capacity of the materials, and it needs to be sufficient to keep the package closed until the point of use. Both methods have some options in the standard, and if you're comparing data, like if you're checking to a manufacturer spec or comparing data from 2 labs, you need to use the same test options to get an accurate comparison. For a peel test, you cut a coupon from the seal. The coupon width depends on the standard you're following. The coupon's placed on a tensile tester and peeled apart to measure the peel force. Once you have accepted criteria for one type of pouch, you can use that same minimum value on different sized versions of the same pouch, since the peel force is normalized by testing a coupon. If you run a burst test, the whole package is filled with air and pressurized until it bursts. Burst acceptance criteria is dependent on material and size, and to a degree, the shape of the package. So each package may need a different specification, depending on how great that burst pressure difference is. Now, let's look at a video with some examples of seal peel testing. So we would cut a portion of the seal, we pull it apart on the machine, and we measure the force required to separate it. Here's a graph. The maximum force would be the highest value on the curve. The energy is the area under the curve, and the average force is the average force from the middle. This is a typical peel. This is an example of elongation. And this is an example of delamination. You can see the material layers have separated in both of these examples. When your seal isn't meant to be peelable, sometimes you'll try and peel it apart, but you'll get a material break. Now let's look at the burst strength video. So if you have a porous package, we need to seal those to help us to pressurize the package. We're going to attach it to our tester, and we're going to inflate it until it bursts. And our tester will record the maximum pressure. And then we need to determine where the package bursts, because we also have to report the burst location. Now, sometimes if you have a seal that's not designed to be peeled open, you may end up with a package failure, which again, neither of these are bad. That's just showing the package doing what it was intended and designed to do. Now let's look at some integrity options. Integrity tests are intended to make sure that there's no breaches to the sterile barrier that could render the package non-sterile. Here are the most popular options are bubble and dye. Both these are pass-fail or variable data tests. As such, the acceptance criteria is normally that they all pass, though theoretically there are some sampling plans that do have allowable failures for very low risk situations. And those usually have very high sample size numbers as well. This will all depend on how you write your sample size justification. The integrity component is perhaps the most vital one, and it would be very hard to justify less than 100% pass rate for a sterile package. For a bubble test, the whole package is tested, and the method can reliably detect a 250 micron defect. The test is run by inflating the package underwater to a set pressure and looking for air escaping as bubbles. For the dye test, only the seals are tested, and this method can reliably detect a 50 micron defect. The test is run by placing the dye on the seal and visually looking for it to pass through a channel. Let's look at the bubble video. And here we can see some examples of what a 250 micron defect would look like. We'll put it in the tub, we'll inflate it to that pressure, and we'll look for bubbles. Now here is a channel leak, and this is a leak. This is a puncture leak here in the film. And now we have one down in the Tyvek. Now, when you have a porous material, since it's breathable, you can get bubbles coming through the package, and we usually will use a lower test pressure to try and minimize that. When you have a label, you can see that we look like we have streams of bubbles coming from single points, but this is actually just because the label is there. This is a fold failure. You can see there that at the fold location, we do have bubbles escaping, but they're not escaping at quite the same rate. So don't fold your packages. Now let's look at the dye migration video. Here we're going to look at a couple of different options that we have for dye. We have injection, edge dip, and non-porous. Here's an example of the injection method. We just inject the dye into the package, and then we're going to rotate the package around so the dye touches each seal. And we're going to look for dye passing through the seal to show us that there's a channel. Okay, here's an example of a good seal, and this is a channel. That's a 50 micron wire. If we're doing the edge dip, we just dip the edge of the sample into the dye, we pull it out and look at it. This one here is a good seal. And here's, look how fast this channel appears. It's almost instantaneous. Here's the non-porous method. We're going to inject the dye inside, but since we can't visually see the dye coming through, we'll blot it against a paper to help us to visualize. Here's a good seal, and then there's the channel. And again, these are 50 micron channels. Microbial barrier testing is intended to verify that microbes will not penetrate the material or package under normal conditions. Now, which test is best depends on the packaging materials and package type. Both F1608 and the Gurley non-porous verification need about a 2 by 2 inch section of material. If your package is smaller or special, you may need to consider a custom test like aerosol challenge. F1608 bombards the material with Bacillus atrophius spores under exaggerated flow, so that there usually will be some penetration that can be measured. The number of spores that penetrate is compared to the number on the positive to calculate a log reduction value, which makes this a variable data test. Gurley non-porous verification uses a Gurley porosity tester to confirm that there's no significant passage of air over a 1 hour time block. Per ISO 11607, a demonstration that the material is non-porous will satisfy the microbial requirement. This is based on the idea that if air can't get through, then neither can microbes. And the Gurley is a pass-fail test. The aerosol challenge is a custom test where whole packages are placed in a chamber and lightly showered with Bacillus atrophius spores. The packages are aseptically opened and the contents removed and checked for the presence of bacillus using sterility test techniques. As you can imagine, it's not easy to open a package covered with an indicator organism without getting cross-contamination. And the test is dependent on the probability of the organism landing in just the right location to be detected. So there are some limitations to this test. Aerosol challenge is also a pass-fail test. Now let's look at our test videos. Here is the F1608. Okay, this is for porous materials only. So we can't use non-porous materials like forests or films. And because we're required to have a 47 millimeter disc, we need to make sure that we have an area that is not including the seal and not including any labels. And you can see here with my identical packages that it may make a difference how your labels are placed. We cut a 47 millimeter disc, and then we're going to put it in our test apparatus. We can accommodate up to 5 samples in here, but we're only required to test 2 for the standard. We're going to use a nebulizer to create an aerosol, and then we're going to run this at a set flow through our test system. We'll collect what comes through, we'll enumerate that, and we'll take the log reduction value of the positive versus the samples to give you that log reduction value. Now let's look at the Gurley. This is a very simple and straightforward test, and it's for non-porous materials. We're going to take this material, we're going to put it in the tester and clamp it. And then we're going to release this cylinder and measure the distance. Now, if this were a porous material, that cylinder would continue to drop until it reached the bottom. We're going to measure this distance again after an hour. And if the difference between the 2 measurements is less than 1 millimeter, then the sample passes. If it's greater than that, then that probably means that either we didn't do a good job measuring, or the sample had a slight porosity to it and the cylinder dropped a little bit. Well, let's look now at this aerosol challenge test. This is going to test the whole package, not just the material. We have a customized chamber. We're going to put them in the chamber and space them out to expose as much surface area as possible. And then we're going to use another nebulizer to aerosolize the Bacillus atrophius so that it can distribute throughout the chamber. We're going to monitor our chamber using Anderson samplers to get a mean particle size. Once we've got the samples exposed, we need to do that sterility component. We're going to disinfect the package, and we're going to open the contents aseptically, and we're going to put it in a growth media. And this is an example after incubation of what Bacillus looks like versus a non-growing sample. So that's the aerosol challenge test. Now that we've reviewed the most common tests for each of these 3 categories, let's see which ones are most appropriate for which types of packaging. For strength, either the burst or peel are used. The selection mostly depends on what history you have at your company. If your company uses peel testing for other devices and has an established peel specification, well, that's probably the best route for you. Likewise, if you have historical data on burst, then that would be the way to go. Setting the strength acceptance criteria is probably the most challenging part. If this is your first time and you don't have any history with the packaging, the pouch supplier may be able to give you some guideline specifications. And if you use those, then you'd want to repeat whatever test they have data for that you're comparing to. For the integrity test, there is a preference towards bubble testing. The dye migration test is more sensitive, but it only looks at the seals. And most of the packaging failures will actually occur in the packaging material as a result of interactions between the product and the packaging during distribution. This is usually some type of breach or hole, and the dye test doesn't look for that. If you have a container type package, then there are more sensitive options available using container closure integrity test methods, and we have a few other webinars on our website specifically on CCI test, and so I would encourage you to check those out. For the microbial barrier, the choice really depends on what the packaging is made of. If you have a porous component, usually Tyvek, as part of the package, then your best bet is ASTM F1608. If your package is entirely made of non-porous, flexible materials, The ASTM 2981 Gurley non-porous verification is your best bet. It's always preferred to use a standard method from a standards organization. These methods have a set procedure that's been tested in multiple labs and that notified bodies are familiar with and have confidence in. Always use a standard test when possible. However, if you have a special design, something that's not a bag, a pouch, a blister, or a tray, or your package is too small, then you may not be able to use those standard methods and will be forced into something custom like an aerosol challenge test. Custom methods should only be used as a last resort when the package can't be tested any other way. Family grouping is a concept that's used in many aspects of medical devices. When you have multiple products that are similar, you can group them into a family, and then you test a representative of the family and apply the validation to all members of the family. What makes a good family will depend on what you're using the family for. If you're creating a family for sterilization, you'd be looking at things about the products that make them similar for sterilization. For packaging, you're looking at what makes them similar from a packaging perspective. Now, sometimes these may end up being the same products at a family, and sometimes you might need to create a different family for sterilization than your family for packaging. Let's look at some of my examples in my slide here and see how we can make some families to illustrate this concept. I have a monitor and an identical monitor with an accessory. Well, I think these might fit well together. And there's a larger monitor, which may be a good fit since it's pretty much the same but larger. Oh, what about this projection screen? It also shows a picture, but it's a bit larger. Is the size too different? Oh, and does it have the same electronics? Because it's a projection screen instead of a monitor. I wonder if that makes it too different. I also have a typewriter, a printer, and a fax. All produce paper documents, so they might be a family. Or since the typewriter is a legacy product using a different printing mechanism, maybe it doesn't really belong here. What about putting all of these products together into a large family because they're all electronics? Well, that could be done, or it could be multiple smaller families. It all depends on the justification and the rationale used, and that's really my point. Whatever reasons you have to consider these similar, you need to document and explain in a logical way that makes sense to a reviewer. And this is my parting thought on family groups. While grouping is a great way to save on validation costs, you have to remember that the family sticks together. If there's a problem with one product, the whole family is affected. And if you need to put a large family on hold while a problem is resolved, it could have potential consequences to your validation and to your business. Now, once you have your family, the next step, oops, a little too far. The next step is to select a worst case representative that you'll validate. As with family grouping, the rationale that you use for what's worst case will depend on what you're specifically looking at. For packaging, we look at things that present the most severe challenge to the packaging, which may or may not give us the same product if we were selecting a worst case for sterilization. Here are some items that would be worth considering when selecting a packaging worst case representative. What device in the family is able to cause the most stress to the seals during sterilization? And this relates to the vacuum pressure levels in some sterilization cycles. Which device is the heaviest? This also relates to stress on the seal. Which device has the most sharp edges or protrusions that could potentially create holes in the package, either directly or through abrasion during transportation vibration? How are the devices packaged? Do some have more or less protective packaging than others? How does the device size compare to the packaging? Improperly sized packages can cause more stress to the package. How are the devices stored? Is one set of conditions more severe? This can become quite challenging if you don't have a sound understanding of what impacts different things can have on packaging. You not only need to make a logical selection and back that up with evidence, You also need to write a good justification document that explains the why in a way that's easily understood and makes logical sense. Again, family grouping and worst case justifications are a service offered by Nelson Advisory Services, not just for packaging, but for sterilization and cleaning as well. Well, let's review. We made our plan by selecting appropriate test options and a statistically valid sample size and wrote it all up in a validation protocol. Then we may have done some family grouping and worst case justification to reduce our testing burden. And well, now we're ready to start testing. And this is a good time to talk about making samples. As we learned earlier, samples should be in the final configuration and final packaging with all intended accessories, including the IFU and protective packaging. We want to make these validation samples in a worst case condition to add more robustness to the validation. This includes using the low parameters for sealing and a 2x worst case sterilization. Now it's time to send them to the lab. So let me give you some helpful hints for success. The shipping boxes are the test samples. You don't want them to arrive at the lab already damaged. So you should protect them either by using an overpack box or maybe a pallet configuration. You need to label the boxes carefully so it's clear which box can be opened and which needs to be tested. The first picture in the slide comes from FedEx, and it has both an example of overpack and good labeling on the boxes. Remember that multiple people are going to touch this sample at the lab, and at some point, the overpack will be removed. So it's important to label both boxes. The second picture is an example of a label that indicates to the lab that this box can be opened to look for paperwork, and that this box must be opened to get to the real test box for testing when the time comes. The last thing you want is a delay at the lab for testing the overpack or accidentally opening the test box and then needing to have it resealed. Well, that ends this packaging validation 101 according to ISO 11607. This slide has my contact information for anyone who needs it. Now, I talked today about several services and documents that Nelson Labs Advisory Services can offer in relation to packaging validations. Nelson Labs and Sterigenics, our sister company, also offer webinars and other educational resources on a number of sterilization and medical device and pharma testing topics. So if you'd like to learn more, please check out those websites. And with that last piece of business, I think we can move on to our question and answer session. So I'm going to turn it back over to you, Mike.
Well, thanks. Thank you, Jennifer, for that thorough and informative presentation. We certainly are happy to have you share this knowledge with us. We do have a few minutes to take some question and answers, so we'll just get right into that. So here's our first one, Jen. What considerations should you give to the glass transition temperature, TM, of polymers when choosing temperatures for your accelerated aging? What if you have met multiple polymers with your various TG in your device?
Well, when you are looking at glass transition points, you want to go with obviously the one that's the lowest. So you would look at all the polymers that you have, figure out which temperature the one with the lowest temperature tolerance has, and you would want to be probably maybe 15 degrees centigrade below what that glass transition temperature was, just to make sure that you don't experience any catastrophic events happening during aging. You wouldn't want to melt any of your product, or because at that point none of it's usable and you'd have to start over. Most of the packaging materials that are common in the industry are capable of that 55C, but not all product is, and so you should definitely consider that when developing your aging plan.
Great, thanks, Jen. Okay, here's our next question. Is there any standard for the climatic stressing?
There are standards for conditioning. I don't think they're specific for climatic stressing, but the ASTM D4169, which is the standard that we are following from ASTM for distribution testing, does have a section in there about the climatic stressing. We mentioned the 3 common ones that we do, but there are other options, and really understanding what your distribution cycle is would help you to select some alternate ones. If you had a really atypical distribution cycle, or you wanted to challenge your samples, there's like a dozen different stressing conditions that you could choose from. The other standard that's very common is ISTA, and they also have about a dozen different temperature things in their climatic stressing portion of their document that you can choose from. So there isn't a separate standard, but it is included in the distribution standards.
Great. Okay, here's our next question, Jen. Is there any acceptable justification or conditions for not performing microbial barrier testing? We use a tray sealed with Tyvek lid, and we have never performed microbial barrier testing, and this device is approved by FDA.
Well, some of this may have to do with timing. Remember, ISO 11607 was first published in 2006, and it really, there weren't any guidelines before then. 11607 is very specific on saying you need to address microbial barrier, but it doesn't say exactly how. If you have a really old validation, it's possible that somebody's looked at it using an older standard that wasn't specific for including this element. But there are multiple ways to do it. We usually advise people to just do the testing because it's very easy to do if you're running the validation. But another option would be to just do it based on writing a justification on published data. If you have a Tyvek component, you can go to the DuPont website, and they have their reference guide for Tyvek, which is full of a lot of test data that they accumulated and have published. So it would be available for you to use in a document like that.
Perfect. I think we have time for one, maybe two questions. So let's go with this one. Should sterility testing be considered as part of packaging validation?
No, we don't recommend sterility testing for any package testing. Sterility is not part of a packaging validation. You'll notice that even though we've got a microbial component, the bulk of the validation is really geared towards physical testing of the barrier to show that the barrier meets all the acceptance criteria of being a sterile barrier. And really that's one of the great things about having ISO 11607 is it makes that very clear that like this is how you should validate your packaging, and this is the concepts that you should use. So we know we've seen people who try to send samples in for sterility testing after they've been aged, and they hope that that would work. But the sterility test, it's one test, it's not very sensitive, it's looking at a very small sample size, and if you get a positive in that test from contamination, then where does that put you? It throws everything that you've got going on into question. So we do not recommend sterility testing for packaging.
Great. Okay, I think this is going to be our last question, Jen. How soon after accelerated aging one should perform real-time aging? Can they be run concurrently?
Well, ISO 11607 says specifically that they need to start concurrently, and it defines that as within 3 months of each other. So they are two separate things, and you're not required to do the accelerated aging, but you should have documented evidence that they are both running. And mostly that's because the accelerated aging would get done, and then nobody would remember to go back and do the real time. So they put that in there to get them done concurrently. That way, you didn't have to worry that 2 years down the road, somebody says, oh, wait, I thought we were supposed to do an aging portion. And at that point, you're kind of in a lot of hot water.
Great. Well, thank you, Jennifer, and for this presentation and for all of your insights. And I'd like to thank you, the viewer, for attending this session. This session will be available on the Nelson Labs website in about 3 to 4 days. So look for this next week if you would like to rewatch this or share with your colleagues. We hope that you found this information quite valuable. Again, thank you for attending, and we hope you all have a great rest of your day.